Hello everyone, I am Chatya from 2nd MSc Microbiology. In this video, let's discuss about microbial production of Hepatitis B vaccine. Hepatitis B and vaccination. Hepatitis B is a viral infection which attacks the cells of the liver. It can be very serious, causing chronic liver failure, liver cancer and death. Hepatitis B can be prevented by a vaccination. The first vaccine became available in 1981. A course of 3 vaccine injections are given, with the second injection at least 1 month after the first dose and the third injection given 6 months after the first dose. The vaccine contains hepatitis B surface antigen, that is HBsAg. It is produced by yeast cells into which the genetic code for hepatitis B surface antigen has been inserted. Recombinant hepatitis B vaccine. One of the most recent developments is the production of a vaccine against hepatitis B using genetically modified yeast cells. The gene coding for the hepatitis B surface antigen is isolated and cloned into the vector under the control of a strong promoter. The cloned gene is then transferred to the yeast expression system. The gene is allowed to express in the yeast and the recombinant protein product of hepatitis is obtained. The protein is then later purified and used for vaccination. Microbial production of recombinant hepatitis B vaccine. Step 1. Gene selection and isolation. The hepatitis B surface antigen gene is selected for its immunogenic properties. The DNA sequence encoding hepatitis B surface antigen is isolated from the hepatitis B virus genome using molecular biology tools like restriction enzymes and PCR. Step 2. Cloning of the hepatitis B surface antigen gene into a yeast expression vector. A yeast expression vector, example YEP, YCP or YIP plasmids is selected. These vectors contain a strong yeast promoter, example GAL1, ADH1 for I expression, a yeast compatible replication origin or integration site. The hepatitis B surface antigen gene is inserted into a vector using GNA ligase. The vector includes a selectable marker gene, example URA3 or LEU2 to identify successfully transformed yeast cells. Step 3. Transformation into Saccharomyces cerevisiae. The recombinant plasmid is introduced into Saccharomyces cerevisiae cells using the lithium acetate. The yeast cells, when transformed, are capable of producing the hepatitis B surface antigen protein. Step 4. Cultivation and fermentation. Transformed yeast cells are cultivated in a complex fermentation medium containing glucose, carbon source, nitrogen and minerals. Yeast cells multiply and express the hepatitis B surface antigen protein in fermentation tanks under controlled conditions like temperature 30 degrees Celsius, pH 5 to 6. The cells are cultured to high densities while optimizing growth and protein production. Step 5. Harvesting of yeast cells. Once the yeast cells have sufficiently expressed the hepatitis B surface antigen protein, the cells are harvested by centrifugation or filtration to remove the fermentation rod. Step 6. Disruption of yeast cells. The yeast cells are disrupted to release the hepatitis B surface antigen protein. This is achieved by mechanical disruption methods such as bead milling or enzymatic methods like zymolysis digestion. Step 7. Purification of hepatitis B surface antigen. Hepatitis B surface antigen is purified through a series of physical and chemical steps: centrifugation, chromatography and ultrafiltration. Centrifugation, which removes cell debris and separates the protein-rich supernatant. Chromatography techniques such as ion exchange or affinity chromatography are used to isolate the hepatitis B surface antigen protein from contaminants. Ultrafiltration concentrates the antigen and removes small impurities or unwanted proteins. The purified hepatitis B surface antigen protein is stabilized in a phosphate buffer which maintains the pH and ionic balance to preserve the protein structure and function. In some processes, formaldehyde is used. Step 8. Virus-like particle VLP assembly. The purified hepatitis B surface antigen proteins naturally assemble into virus-like particles in a phosphate buffered system. Virus-like particles allow the immune system to recognize them as foreign and trigger a response. They are non-infectious as they lack viral genetic material, ensuring safety. This self-assembly process enhances the immunogenicity of a final vaccine, providing effective protection against the hepatitis B virus. Step 9. Formulation. The assembled virus-like particles are formulated into the final vaccine preparation by adding adjuvants such as alum, that are aluminum hydroxide or aluminum phosphate, to boost the immune response. Stabilizers such as phosphate buffer may also be added to maintain the antigen structure and activity. The final vaccine formulation is adjusted for isotonicity. That means matching the salt concentration to that of the body fluids to ensure compatibility with the human body. The vaccine is tested for sterility and safety before proceeding to sterilization, filling and packaging. Step 10. Quality control. The bulk vaccine is tested for purity, efficacy and safety. Residual yeast proteins and endotoxins are carefully removed to acceptable limits. Step 11. Sterilization and filling. The final vaccine is sterilized by filtration to remove any potential contaminants. It is aseptically filled into vials or refill syringes and sealed. Step 12. Packaging and distribution. The vaccine is labeled and packaged under sterile conditions. It is stored at 2 to 8 degrees Celsius and distributed using cold chain logistics to maintain its stability. Here is a pictorial representation that briefly explains the entire production process of hepatitis B vaccine. Gene is isolated from hepatitis B virus and is inserted into the plasmid DNA using restriction enzymes. Recombinant DNA is formed and is inserted into the yeast cell. The recombinant yeast cell is cultured in fermentation tank. Recombinant yeast cells multiply and produce hepatitis B surface antigen in fermentation tank and is extracted and purified, and finally the hepatitis B vaccine is produced. Risks of hepatitis B vaccine: soreness, redness or swelling at the injection site, mild fever or fatigue or some of the mild symptoms that may occur after vaccination, hypersensitivity or anaphylaxis due to vaccine components. Extremely rare cases of Guillain-Barré syndrome have been associated with vaccines. Autoimmune conditions such as multiple sclerosis is linked to hepatitis B vaccine. Safety of hepatitis B vaccine: highly effective in preventing hepatitis B infection with over 95% protection in healthy individuals. Extensively tested in clinical trials demonstrating safety across age groups. Severe reactions are extremely rare, making the vaccine safe for the general population. Safe for use in infants, pregnant women and immunocompromised individuals. Recombinant vaccines have killed form of strains, eliminating the risk of infection from the vaccine itself. Supported by World Health Organization, Centers for Disease Control and Prevention and other health organizations for incorporation in immunization programs, recombinant technology reduces the risk of contamination or transmission of other diseases. Thank you for watching.